So hello everybody, we are talking about the sound localization and auditory scene analysis and how the brain processes the signals. Those are important for auditory scene analysis. Uh, and we were talking about the superior olivary neurons, medial superior olive and lateral superior olive, and those two structures have the neurons. They can calculate the interaural time difference and interaural intensity difference. So they are processed separately. Okay, and then as the signal goes up, they converse together. Okay, so time and intensity pathways, they join together and create a map of space. So combining those, it gives a map that has the neurons. They just specifically tell the exact location, how many degree in azimuth and how many degrees in elevation. So those neurons are definitely bimodal neurons, and they can immediately tell the exact location. So this auditory space map is located in the area which is called the optic tectum. Okay, so this is optic tectum, where you have the auditory space map. So these neurons can exactly tell you the location of the sound source, and also these neurons receive the signal from the visual neurons. So you see here, auditory and visual are combined. So when we hear a sound, like somewhere in the right side of your body, a dog barks, then you listen that, and also at the same time you move your head to look at that dog. So two things are combined, right? Your visual space information and the auditory space information. So you see, at the same time you listen, and those two must combine together to give you the perfect sense of the location. So visual is coming from this way. If visual, like 20-degree azimuth, 20-degree elevation, also 20-degree auditory azimuth, 20-degree auditory elevation. Okay, so these two and these two both are joining here to give you the precise information, uh, sufficient information about the location of the object. Now we will talk about another type of auditory scene analysis that I mentioned, that echolocation, right? So echolocation is different than the sound localization we do, or the owls do. In echolocation, the subject sends a pulse, that means a sound, and that sound will hit the surrounding objects, and the echo that return uh, would be analyzed by the auditory part of the brain of the echolocating animal. So most powerful echolocators are the bats. other creatures, for example, dolphins, or whales, elephants, or some other creatures, they also use echolocation. And the bats are the most, diverse mammals on the earth, and more than 1,000 different species of bats have been, identified. So just think that a big part of the, mammals are actually the bats. And um, among those, like, more than 1,000 different types of or species of bats, we divide them into two groups based on the ability to use their echolocation. So we have echolocating bats and non-echolocating bats. Echolocating. So non-echolocating bats are like birds. Okay, for example, fruit-eating bats. There are many different, names of bats. You will see nose-leaf bats, fruit-eating bats, fox bats, or vampire bats. All those interesting names you will see, and some of them use echolocation, not all of them. Okay, and another way to classify the bats is by their food habit, eating habit. Some bats, they eat vegetables or fruits, and some bats, they eat meats or blood. So we can also classify them that way. However, in neuroethology, we usually classify the bats into two types, uh, echolocating and non-echolocating. Now, echolocating bats are classified into uh, FM bats and CF FM bats. Okay, so the difference is FM indicates frequency modulating. So how the bats call, based on the calls we classify into frequency modulating, and CF is constant frequency. Okay, and if you see frequency modulating calls, if you record the call, if this way is the frequency and this way is the time, this is frequency. So frequency modulating will be like, sweep like this. So the frequency is changing, you see, going down from high to low. So broadband, we say FM, because the frequency is changing or modulating, going down, and this is broadband, right? The frequency band is broad. Radio frequencies, we say FM band, broadband. Some bats produce the calls, those are FM or broadband calls, and constant frequency is the frequency doesn't change over the time. So this is fixed, constant. Okay, so frequency, you see the frequency is not changing. So narrow band, CF, narrow band. Okay, sometimes we see that CF bands add a small part at the end, that is FM. So we will say CF FM. This part is constant, this is modulated broadband. Okay. Now the bats produce click or pulse using their mouth or nose. Okay, and sometimes they use both together, and the eyes of the echolocating bats are pretty small, and they use their eyes much less than the sound. However, non-echolocating bats, their eyes are bigger, and you will see they um, use their eyes to like catch the prey, or find the food, or fly, find the ways. So they use their eyes, but echolocating bats, they don't use their eyes a lot. That's why their eyes are smaller. But if you see the echolocating bats, their ears are pretty big, large pinnae. So the pinnae is pretty big. Okay, to capture the sound. In some echolocating bats, you will see nose leaf, a cartilage is present in the nose, and that actually helps them to present the pulse precisely in the right direction. Anyway, so when the bats fly at night, usually they come out at night. The reason is that they don't need to see, they use echolocation. So at night it is easy for them to find the prey. During night many insects, they come out. During the daytime they don't come out, so at night the bats can find their prey easily. Also, you know that less competition, right, at night time. So all those things help the bats to come out and find their prey at night time. When the bats come out, you will see that thousands of bats, they fly together. And if you see the colony of the bats, you will see the bats stay together like clusters. I had the opportunity to catch the bats. We used to go to catch the bats for our research when I worked with Dr. Suga. We used to go to old churches, or abandoned houses or stations, or caves. In Missouri we have so many caves, and if you go inside the abandoned caves, you see millions of bats are clustered together in the ceiling. So it's very interesting how they live together, close to each other, and they come out together too at night. And the pulse and the returning echoes, these are the components they use to analyze and get the information about the object. Now, by analyzing the pulse and the echo, they can detect a number of important information about the object. They can detect the distance, detect the size, texture, velocity. So flight to flight information. Bat is flying, the prey is flying, and by analyzing the echo, the bats can also determine the velocity of the prey. Even if it is not stationary, they can update the information. So it is very powerful, their auditory system is extremely powerful to get all those information by analyzing the sound returning echo. Okay, and we already know that bats generate ultrasound, which is uh, much higher than human audible sound. In most of the cases, bats use ultrasound, so we don't hear that. And as I mentioned, that use mouth and nose, and the sound echolocation calls or pulse range between 14 kilohertz to 100 kilohertz, and you know that our maximum limit is 20 kilohertz. So most of the sounds they make for echolocation is beyond our audible sound or capacity. Uh, we can use the tools or equipment that can record the high frequency sound. So when I used to work on the bats, we also used to use the bat detectors. It's like a transistor type equipment that you can carry and go to an area where you think the bats are living, and you can turn on that equipment detector, and it can pick the high frequency sound that we don't hear. So it also can tell you what kind of bat that could be, because different species of bats have different types of sounds. As I told you, FM, CF, FM, CF bats. So if it is CF sound or FM, by uh, recording that, we can also tell what kind of bat is in that area. And we have a library where we have the list of all different frequencies produced by different types or species of bats. So that helps us to detect the bats in that area. So here you see that this this bat is flying towards this object and sending the pulse. So the pulse would hit the object and echo the green lines, the waves return. Now, a term that is called Doppler shift. What is that? The sound waves can be compressed or expand. So when the bat is moving towards the object, getting closer, and producing the pulse one after another, as the bat is getting closer to the object, compression of air takes place, and that increases the frequency of the sound. So as you approach the object, the frequency increases, the frequency of the echo increases. Makes sense? So you send a pulse, and then before the echo returns, you already moved more forward, right? A little bit more forward. So when you will receive the echo, the frequency will be slightly higher. If you go away from the object, expansion occurs. That means you send the pulse, and when the echo arrives, you already are away from the previous position. So the frequency decreases, the sound waves length increase. That means the frequency decreases. So compression will increase the frequency, and that means that you are approaching towards the object, and opposite is you are going away from the object, so the frequency will decrease slightly and the wavelength will increase. Okay, that is called the Doppler shift, and bat uses the Doppler shift as an indicator that if it is getting closer or getting away from the object. Now, um, it becomes more complicated when the object is also moving, right? If the object is stationary, it is rather easier. However, most of the cases the uh, prey are also, like, insects are moving or flying, and that makes the process more complicated. However, bat's brain is extremely sophisticated to process flight to flight information too. Anyway, so here you see um, the calls and echo. Here you see the compression, the frequency increased in returning echo. So here you see the phases, how the bats produce pulse at different phases of approaching the prey. First, when they're looking for, they're just looking for food or prey, they produce few uh, pulses in one unit of time. So you can see here that is called the search phase. That means bat is just producing pulse and waiting to listen the echo, and it will not produce another pulse until the echo returns. Otherwise it will um, hit the returning echo before it arrives. Anyway, so you see bat produces pulses, okay, and listen the echo, and then if it detects something, that some prey, the presence of a prey, it will then move towards it, right? So that is next uh, phase, and in this phase bat will increase the number of pulse, okay, and you see as the bat moves close and close to the prey, or the food, it gradually increases the number of pulse. And it makes sense because they don't have to, as they go closer, they have to wait less uh, amount of time to produce next one, because the arrival echo will be faster than when it was away from the object. So when it gets closer and closer to the object or prey, it will increase the frequency of pulse, and the last phase is called the terminal phase. Here you see the bat is very close to the object, so it needs more precise information. Initially it just needed to determine the location and move towards it. However, as it gets very close, it needs more specific information like shape, size, right, and exact velocity, all those information, texture. So it increases the number of pulse a lot, and up to date the information in a very high frequency, which is very important to precisely catch the prey, and that is the terminal buzz is the last step when the frequency is very high, and just before the bat catches the prey. This is something interesting. If you see the skeleton of a bat, you see the finger bones are so long, and that makes their wings very long, and this is the skin in the wing, which really can spread a lot and help them to fly without spending a lot of energy. Okay, two things. One is the light skeleton. The skeleton is very light, and the large sized finger bones or phalanges uh, make the wings broad, and that also helps them to fly without spending a lot of energy. Also, the bats, when they are in search phase, that means they are just flying and looking for the prey, they use the rate of pulse same as their respiratory rate. So when they breathe, like expire the air, they produce the sound. So they can produce the sound without spending more energy. Just when they breathe out or expire the air, at the same time they produce the sound. So that way they can save the energy. Before the scientists discovered that bats use echolocation, they are very puzzled. They didn't know how the bats locate the prey so specifically. Uh, some people thought probably they have something special in their skin of the wings, some special sensors. Nobody had idea that bats produce pulse and use the echo to localize the prey or sound source, localize the prey or the food. So many different ways they tested that. One interesting thing they did when many people started to believe that something is present in their wings, that's how they can detect the prey. They used library glue, a special type of glue, to cover the wings with that glue and let the bat fly to see if they can still find the prey precisely, and they were able to do that. That means there is nothing in the wing somewhere else. And first the scientist who discovered that bats use very high frequency pulse that we do not hear to localize the prey or find the prey. He was a graduate student. His name is Griffin, and Professor Griffin, he was still a graduate student at Harvard, where he was working on the insects, the sound of the insects. And in the next lab there was a faculty who became very famous. He first invented the equipment that can record high frequency, and he used that to see while the bat flies if they produce any high frequency sound. And he was very excited because he first discovered that bats use high frequency sound to use echolocation, and that's why we didn't hear that sound. So that was the first finding that bats use high frequency sound for echolocation. And then he did many interesting experiments, and as you know, Professor Suga, who I worked with, was one of his students, and he continued his research on the bats. And a couple of interesting experiments. One is that Uh, Here you see this is Griffin, who first discovered that bats use echolocation, and this is Professor Suga. I worked with him, and this is me, interestingly, at WashU biology lab. Around 1997, I used to work with him. So, uh, uh, Professor Donald Griffin, he did a number of interesting experiments. One is he got some strings used in the guitars, the musical instrument, thin strings. He hung them from the ceiling in a hall room and then let the bats fly in the dark room, and he recorded the movement of the bats, and he found that the bats can avoid those strings. Those were extremely thin and were placed, like, very close to each other all over in that hall room. However, the bats were able to, without seeing, avoid those strings and fly in that hall room. So that tells that the bats can very, precisely detect the shape, size, and distance of the object. That is something interesting. And another interesting experiment he performed, to see if the bats can detect the water surface, because when the bats fly at night, some bats they eat fishes. So or when they need to drink water, they need to make sure by listening the echo that that is the water surface. So he also performed the experiments, and inside the lab, glass plate on the floor, and let the bats fly, and noticed that bats thought that the glass surface is like water plane surface, and they used to fly like this to drink water and go up like this. So very precisely they just touch with their mouth and go up. So that means by sound the bats can also detect the texture, if the surface is plain like water, or texturous surface. So interesting experiments were performed on the bats. Professor Griffin mostly performed the experiments. Those were like behavioral experiments. However, Professor Suga, he mostly used neurophysiology to find the processing of echolocation in the brain, in the neurons. So we also know in Suga's lab tested, mealworm. Bats love mealworm. We use strings to hang the mealworm in the dark room, and we noticed that how the bats use their wings to first, grab the food and then put very nicely inside the mouth. So if you see the video, I'll post a couple of videos, you will be able to see how bats catch the prey. It's very interesting. So you see the water surface. Now, the difference between CF and FM. FM sound is broadband, right? The bats use the FM sound mainly to detect the distance of the object. Okay. However, most of the information, like the velocity or the texture or the size, those are mainly processed by the CF pulse. So CF pulse is very helpful to hunt in vegetation where you have a lot of backgrounds, and the static backgrounds, when the fly is moving, the prey is flying. uh, Bat, for example, the CF bat is flying in the forest, like in a rainforest you have many plants, so all these are uh, the trees and plants, these are fixed. This is the bat. Okay, and as the bat moves towards the objects, as I told you, that the echo, the frequency is slightly higher. That means the compression occurs, right? So this is pulse and the frequency is here, and when the echo returns, because bat already moved forward more and the frequency is slightly higher, the returning frequency is slightly higher. So that tells you the distance of the objects. One thing is very interesting, when we talk about the pulse and echo, we usually indicate one object, but in nature you just think that how many objects are around, especially in a forest, how many trees and plants are there, right? So all those information are returning at the same you know time, slightly little different, but together. So that has to process that very highly complicated um, echo. Anyway, so from all these objects the sound returns as echo, and the returning echo frequency is slightly higher, and that is the Doppler shift, right? And now if any small prey like an insect is flying here, if the insect moves towards the bat or away from the bat, uh, the bat can detect that. How? Because all these things will be considered as background. Okay, so although bat is detecting that it is getting closer to all these objects, however, once these become background, these become like a static, and this one is moving in that background. So this is the only object it can detect different than others. Okay, others are like changing in fixed ways, but the flying object is you know giving different signal. So that's how the bats can hunt or hunt the prey in vegetation. To do that, the bats need to use the CF, constant frequency, because if the frequency is modulated, changes, then it will not get precise information. So you must remember that CF is like this, fixed frequency, right? So usually echolocating bats use 4 different harmony. First harmonics, H1, H2, H3 and H4, okay, as pulses. So these are pulses at different frequencies. Okay, so low to high frequency. So this is high, this is low. Okay, and now if the bat is moving towards the object, then what will happen? The echo that will return will be, the frequency will be slightly higher. Make sense? So echo, slightly higher frequency for these pulses, okay, or these harmonics of pulses. So pulse and dotted line is echo. Again pulse, echo. Okay, just know that echolocating bats, CF bats, uh, puts more efforts on H2, or uses the H2 pulse and echo to detect the prey, and depends more on H2. We'll talk about that more. Now, inside the laboratory, there are many ways you can study the neurons sensitive to different harmonics of pulse and echo, uh, and very systematically you will find the neurons. Like you have the neurons that are sensitive to specific pulse and echo difference. So this one probably like 100 hertz, this one 200 hertz. So the difference between pulse and echo, uh, different neurons are sensitive to different uh, differences between pulse and echoes. So everything is very systematically, and you have the neurons for all those different uh, uh, timings. So this is a study on the FM band. So you can see that frequency modulated, right? So the frequency is changing in the pulses. So these are the pulses at different intensities, and this is an experiment. You can see here, this is the bat sitting on the platform, Y-shaped platform, and here is the food target. Okay, and loudspeakers to produce sound, and this is another loudspeaker is this one, and this is a loudspeaker to produce sound, and this sound is called jamming sound. And microphones are also placed in the soundproof room, and now uh, you record the pulse, okay, using the microphones. So you record the pulse produced by this bat. So the bat sends pulse, okay, that hits the target. So these are the pulses bat use. Noise, the bat produces pulses, FM pulses. These are the FM pulses at different harmonics, and uh, this loudspeaker is silent. Okay, no noise is being presented. Then the loudspeaker was turned on, and noise was presented. That means, like another, you can think this is another bat next to this bat, because that bats fly together, right? So many, hundreds, thousands of bats fly together, and all of them are producing pulses. So huge amount of noise is present around the bat. So this study is to just see how the bat responds in the presence of noise. So if you think this is another bat which is producing sound, and that sound is now a noise for the bat because that distracts the bat, right? So when you start to present that noise, you see the bats use a strategy by expanding the tail part of FM bands. So you see the tail part, last part is extended. If you just compare with this, these are like short, and this one is much longer, right? So this you see the end part was extended, prolonged. Here it is very long, right? So when a number of bats fly together and each of them is producing sound, the sound is noise for the other bat. So in that situation, they extend the pulse, prolong the pulse, and that helps them to avoid the noise and get the echo more accurately. So this is just an observation how the bats uh, change the strategy in presence of noise. This is another interesting observation that um, bats produce pulses to get information about the prey. However, there are some preys, they also use different strategy to um, confuse the bats, and that is something very interesting. um, you will see some videos showed that is called evasive or turn away from the threat. Okay, so when the insect detects that bat is sending pulse to find them, the insects also produce sound or calls that will change the pulse and the returning echo, so the bats will get lost, will get confused. Okay. Okay, so now you see here FM calls frequency modulated, right? And CF FM, you see FM, CF, FM, FM, CF, FM. Okay, and you can record in the laboratory or outside the calls. So search, phase, approach, and attack. That's the last phase, terminal phase. We talked about that. So the frequency increases. Now, in Suga's lab, what they did, they recorded the auditory part of the brain to see how it is different, if it is, from other creatures. And what they have found is very interesting. They found although bats are very small, most of the echolocating bats are small, and non-echolocating bats are much bigger in size. Some of that could be very big. However, most of the echolocating bats, like mustache bats or small brown, big brown bats, those are very small, and head is especially very small head, and the brain is really tiny. However, inside that brain you will see a number of structures, much more than other creatures. More than half of the bat's brain is devoted for auditory processing. So you'll see multiple areas more than other creatures. So here you can see different areas in the brain of the echolocating bat. In non-echolocating bats you won't find all these structures. Okay, so here you see DF area, FM area, okay, CF area. Okay, and this is the primary auditory cortex. It's comparable to human and other creatures. We don't have all these extra areas, but we have a one primary auditory cortex. And although in bats primary auditory cortex you have also frequency map, that means the systematic organization of the neurons. However, you'll see in the middle of the A1, you have a large area which is highly expanded. That means the narrow frequency range is present in this big area, okay, around 62 kilohertz, and that area is very big, okay. So in the bat's auditory part of the brain, you have different additional structures like DF, DM, uh, and FM, CF areas, as well as the primary auditory cortex, okay. And here you see different neurons respond to different uh, intervals, which is very important, uh, this neuron likes 4 millisecond, this one 6 millisecond, different intervals they respond. Now why you have different neurons respond to different intervals? Because when you send the pulse and wait for the echo, that time between the pulse and the returning echo received by the brain, um, what is the you know time? Is it 4 millisecond or 6 millisecond or 10 millisecond? Those should be detected by the brain, because based on that timing your brain will determine the distance, right? And if the time is longer, that means the object is farther away. If the time is short, object is near. If the time is gradually getting shorter, that means you are approaching towards the object, right? So you have all those neurons, they can detect different uh, you know uh, times of the echo. So the time between the pulse and the echo, and based on that the brain determines the distance of the object. That's why you have the neurons uh, prefer different times. Here you can see same thing that I showed you. This is the primary auditory cortex, and you see the frequency map, different frequencies, of course much higher than human, and this area in the middle of the map is the DSCF, D Doppler shift constant frequency. You know Doppler shift, I explained you, and also constant frequency. And this is the area where the neurons will detect the Doppler shift, the difference of frequency of pulse and echo, okay. So here again you see I told you 4 harmonics, H1, H2, H3, H4. So from low to high, H1 is the lowest frequency and H4 is the highest frequency, okay. And you see the pulse and echo. Pulse is the continuous line, and echo, returning echo, is the dotted line. So you see always echo frequency of echo is slightly higher, the Doppler shift, when, remember, the bat is moving towards the object. If away, then it will be lower, the returning will be lower, okay. So anyway, so in this case the bat is moving towards the object, and you see 4 different harmonics of the pulse. And as I told you, the bats use this one, H2, CF2 more. And this is something very interesting, that the bat sends the pulse or produces the pulse in a way that the returning echo must fall in this DSCF area, okay. So this DSCF area, as I told you, is expanded but narrow range, it is like 60 to 62 kilohertz. So all these neurons in this big area responds to this narrow range, okay, of frequency. And the bat here you see 60. So bat always produces the pulse in a way that the returning echo uh, must be processed within this DSCF area. So if the bat is approaching towards the object, this is very important to understand. If the bat sends the pulse at 60 kilohertz, then the echo will be slightly higher, right, as I mentioned. So for example, this is 60 kilohertz pulse, and this is echo, which could be 61 or 62. So still will be presented within this area. Make sense? Now, if next, how the bat will decide the next frequency of next pulse, frequency of next pulse. If it says that the returning echo is going beyond the limit, like going to 65 and is returning here, that these neurons are being activated by that, bat won't use that. Bat will now reduce the pulse, so to make sure that the echo returns into that DSCF area, because DSCF area is Doppler shift constant frequency neurons are there. Okay, they can detect the information from the echo. They is specialized for that processing of the Doppler shift. So you see a large part of the brain is processing FM, CF and Doppler shift. So this is how the information we gather from our experiments can be used. Echolocation experiment is extremely useful because we can use the echolocation in many different purposes. For example, many military equipment are using the echolocation and Doppler shift to detect the range or velocity of the target or another war, like jet fighter or another moving war vehicles. So very important for military. Also Navy uses to detect the underwater objects. So echolocation can also be used under the sea to detect the object distance, velocity, and the shape, size, all those information. And medical equipment also, we can send pulse and listen the echo from different organs, because inside the body different organs have different densities, right? So that density can also be detected, because when the echo sound will hit a solid structure, the echo will be different than when it hits a soft structure, right? So some organs, for example, cirrhosis of the liver gets very hard, it loses its softness, right? So in that case, the sound that will return will be different than the normal liver. So just one example. So navigation, we all know that echolocation, and recently the cars are being built that can move by itself without the driver, by just sending pulse and analyzing the echo. All those information we get from the creatures, and like especially from the bats. And auto parking, right, is done by echolocation. Improve human ability. Human brain is not good for echolocation. However, we can improve our brain. Probably you have heard the stories that people who are blind from their very early part of life or by birth, they many of them can use the echolocation and improve the ability to echolocate the objects around them. They can move, produce click, learn how to produce click, the pulse, and receive the echo and analyze that echo to get the information about the objects. Boy, probably you have heard Ben. He passed away. He was very, good in echolocating. He learned his eyes were, damaged due to cancer in his very early childhood, and his mom trained him how to do echolocation, and he was able to move, play basketball, and bike on the street using the pulse and listening the echo. So you can improve the ability of your brain and use echolocation. This is Professor Suga. He performed many elegant experiments, like Nobel Prize, deserving experiments on the bats. This is me. I worked with him few years. So I already talked about this evasive action. Insects or prey can use their pulse to confuse the bat or deviate them from them to save themselves. So that can be done. So go over these last few slides, and you will know that how bats use their pulses and analyze the echo, and which information is given by which information from the echo. So the echo that returns into the bat's brain, a number of information is present in that echo. Okay, Doppler shift that I mentioned, explained, that gives the velocity if it is moving away or moving towards, and what's the velocity. Amplitude, that's the angle. Okay, so the pulse, and this is the insect. So is it above or below? What is the angle? So bat can also detect that from the returning echo. Okay. So that helps to localize, right? Delay, that is the range or distance. Of course, bat sends pulse and wait for the echo, and based on the time difference, it can tell the distance. Amplitude and delay, these two together gives the information about the size. Okay, amplitude spectrum, fine characteristics like textures or small parts of the body. Binaural cues, of course, these two we use to localize. Also human brain as well as owls, we talked about that. So 2 ears are used for sound localization. So these are like common. However, these are additional information bat's brain is getting from analyzing the echo. Okay, this is the boy I was talking about, Ben. He was very good in using echolocation and was able to move around, play and bike. He learned how to analyze the echo. His brain learned, and that means you can of course improve your capacity of the brain to use the echolocation. Okay, so make sure you go over all these things. So two important things: auditory scene analysis, human auditory scene analysis, and I talked about the stream segregation, right? Onset segregation, how to analyze to understand the auditory scene. Also, I talked about the continuity of the sound, restoration of sound. Um, then I talked mostly about the complex auditory scene analysis. One is sound localization by the owls, and another is echolocation by the bats. Okay.